Today I'm going to share with you guys an example of using single-cell spatial multiomics to study the molecular, cellular, and tissue level changes that occurs with aging. As well, I'm going to introduce you to a set of bioinformatics tools that we've been developing during this process in order to get at the underlying biology. It's a project that I've been working on for the last couple of years, and I'm very happy to be able to share with you all today. The focus of our study is on the loss of muscle mass and function with aging. Skeletal muscle is responsible for all voluntary movement, posture, breathing, even temperature regulation. It makes up about 30 to 60% of our body mass, and it's a major contributor to our body's energy metabolism. However, with aging, muscle begins to atrophy, waste away in a process known as sarcopenia. This affects roughly 5% of the population over 60 and roughly 30% of people over 80. And of course, a loss of muscle mass and frailty leads to falls, loss of ambulation, dependencies on care, and is a major comorbidity to other chronic aging-related diseases such as cardiovascular disease and cancer. With the aging population of developed countries, it's expected that there will be more people over 65 than those under 18 by 2035 in the US. And so there's an ever-increasing need to better understand the process of aging and development of therapeutics that can possibly extend health span or longevity. Of course, aging is a multi-system, multi-scale, multi-omic process. It affects the organism at every level, from molecular dysregulation, the genomic, epigenomic, metabolic, protein folding, cytokine signaling level. And these changes at the molecular level can propagate into tissue dysfunctions and really multi-systemic deterioration. And really, the only way to comprehensively understand the process of aging, this complex biological process, is to do it with multi-omic studies. One of the aspects of aging at the tissue level is the loss of kind of balance between damage and repair. You know, as our tissues get older, there's increased stress and increased demand for repair. And tissue homeostasis is normally maintained through coordinated efforts of various cell types I've highlighted here. And those cell types kind of work together to repair the wound and reform the function of the tissue. And age-related dysregulations have been observed in various cell types, even within a single tissue, such as muscle. The muscle fibers atrophy, there's increased inflammation, there's fibrosis. The innervation of the tissue also becomes dysregulated. And various aspects of the age microenvironment is going to affect whether the damage associated with tissue stress is exacerbated or even prevent the proper repair of the tissue once it's been damaged. However, thinking about what's been done and looking at these tissues, it's really not been holistically examined at various scales and single cell resolution at what changes with aging. Of course, to get at some of these biological questions, such as can we actually assess molecular drivers of aging in different cell types and identify what molecular mechanisms are part of this process, what changes there are to the tissue's cell composition, the organization of these cells within the tissue, and can we actually identify signaling that becomes dysregulated, and maybe we can target those for therapy. And in order to understand these kind of biological questions, we had to develop, of course, rely on technological advancements and develop new tools to study this process. In terms of understanding multiple distributions of proteins within the tissue, we're here using multiplex imaging, highlighted by CODEX, which is developed by the Nolan Lab and commercialized by Akoya. We've been, in order to actually study these highly multiplex images and high dimensional cytometry data, we really needed to also improve the image processing pipeline associated with that to overcome some of the limitations with microscopy at the current state. So we've developed a new tool that I'm going to introduce today called CRISP, which is able to really register images, deconvolve them, and do it at a higher resolution and subpixel resolution. In order to then take, your images and then get single cell data, of course, we use cell type clustering and annotations. And this can be traditionally done with k-nearest neighbor algorithms, Louvain clustering, community-based clustering algorithms. I'm going to introduce today what I've been calling computer vision assisted clustering, using a new pipeline that we've been working on, which really helps with these not completely high dimensional, with antibody panel selected data sets that already have inherent kind of populations that are going to be highlighted. And then, of course, how do we enrich this data set with transcriptome data, single cell transcriptome data? And you've heard the talks from Kai about Gluer and the use of stevia to align CITE-seq data and multiomic data with spatial proteomic data from CODEX. Today, we've been focusing on kind of the downstream analysis of spatial cell-cell signaling using a new connectomics tool that we're calling SPACE. All right. So I'll start at the beginning at image processing. So once you get your, CODEX or multiplex imaging data, the most important thing is that you need to process this data in a highly efficient and accurate manner. We've been working on a tool called Convolutional Registration of Images at Subpixel Precision, or CRISP, which is a GPU-accelerated algorithm that is able to translate and register images at subpixel resolution. Does this in the Fourier shift domain, and it includes Richardson-Lucy deconvolution, on the fly kind of point spread function generation, and chromatic aberration correction. Here you see a high magnification image of a tissue. You can see that we are kind of away from the perfect focal plane here, so you're getting blurring due to the use of wide field lens and oil immersion. And not only that, due to the setup of the lens itself, you can see that there's chromatic aberration between the red channel and green channel. And of course, if you were to process this data with chromatic aberration, you will get a discrepancy between your spatially localized signals here. And so with CRISP, what we're able to do is not only deconvolve the image and make it less blurry, you can see that we've corrected the chromatic aberration. And this is actually the same tissue slice in the Z-stack that you're seeing. So the resolution and the deconvolution here is really robust. Beyond that, what we're able to do is look at the entire tissue, and when we're stitching, multiple tiles together, here we're in the range of multiple millimeters of tissue. You can see that certain areas of the tissue, it becomes blurry due to possible slants and a slant or slope in the cover slip while it was being imaged. Here we can reconstruct the entire tissue, and you can see that there's a roughly 4 micrometer dip towards this corner of the tissue here. And so with CRISP, what we're able to do is at the subtile and subpixel resolution, Z-shift this entire tissue back into a single focal plane that is really, you can see much more in focus. In this way, you can actually have a single plane of perfectly in focus data that you can work off of for segmentation and quantification as well. And not only that, because we're reducing the number of slices in Z that you really need to keep, since the tissue is now flat instead of a slant, we can actually reduce the amount of data storage that is required to maintain all of these data sets. And of course, the images, the resolution of the image is maintained. And because all the translation and stitching is done at subpixel resolution, you can see that the image is just as high quality and sharp before translation at the subpixel resolution, which normally with other algorithms will require interpolation and kind of blending. So this allows us to look at a tissue, a much larger portion of tissues in CODEX with lots of antibody markers. We've developed a panel of roughly 35 to 40 antibodies to study the different cell types in skeletal muscle. And so this tissue that you're seeing here is a longitudinal section through the muscle of, through a mouse muscle, and it's roughly about 6 millimeters squared. You can see here, we've labeled the muscle fibers in red, the tendon, which connects the muscle to the bone, in cyan. You can see in green where the motor neuron tracks are and where it innervates the muscle. Here's a zoom in of a neuromuscular junction with this very typical pretzel-shaped and motor end plate. We can also observe immune cells in the muscle and vasculature here in white. And you can see that the tissue is really made up of multi kind of lineage cell types, everything from endothelial, vasculature, immune cells, neural to muscle to tendon and fibrotic cell types. To get further insight into other cell types that could come into the tissue during injury, we've conducted a series and a time course of muscles injected with a notexin, which causes muscle fibers to hyperpolarize and die. And this is then cleared by the immune cells. You can see here lots of infiltrating immune cells. And even though we're looking at kind of snapshots in this time course, what we're able to see are regions that kind of are more damaged and are slowly regenerating versus regions that are more further along in the regeneration. So even though we're looking at snapshots, we can actually get a pretty comprehensive understanding of the process of tissue regeneration. And so here you can see actually newly formed muscle fibers that are being reinnervated while other regions are still kind of less further along and are being cleared by immune cells. To even go further, we included antibodies that delineates functional subsets of muscle stem cells and, of course, immunophenotyping markers, so that we can look at, the progression from a muscle stem cell here in green in the upper left panel to MyoD positive transient amplifying progenitors or myoblasts in red in the second top left panel, to differentiating progenitors, myogenin positive myocytes that are in green, that are in yellow, and then early differentiation markers, newly formed myotubes that are positive for embryonic forms of myosin in cyan. That's also in the top right panel. We can also look at the interactions between these stem cells and immune cell types by including antibodies for immunophenotyping. So CD45 labeling all immune cells, macrophages being labeled by CD48 in green. You can see how close in proximity these macrophages can come into the myogenic progenitors, their physical and distal interactions here. And you can see even rare cell types like CD3 positive T cells that are infiltrating muscle during regeneration, and symptom-dendritic cells as well in the bottom right panel here in cyan. So how does this translate to studies of aging? So while I was working on CODEX, another postdoc in the lab identified that prostaglandin signaling is lost and reduced in aged muscle due to the upregulation of a prostaglandin degradation enzyme called 15-PGDH. So by comparing mass spec levels of PGE2 and PGD2 between young and aged muscles, she identified that there's a significant decrease in the amount of prostaglandins in the tissue, and that is directly due to a increase in the amount of this degradation enzyme called 15-PGDH, as shown by qPCR for its mRNA levels. And when we knock this gene down, this degradation enzyme down, you can actually restore the amount of PGE2 in the tissue, in the aged tissue, as seen in the top right panel here. What was really amazing is that just by knocking down this one enzyme in aged muscle, within a month, the aged animals were actually regaining their muscle mass. So observed a 15 to 20% increase in the muscle mass, but also an increase in the muscle force. And these mice were able to, and we were also able to confirm that with a small molecule inhibitor, that the mice are able to be stronger and have regained muscle mass. And what's really striking is that overexpression of this enzyme in a young mouse model using AAV was actually able to cause a deterioration in the muscle and muscle wasting within a month. However, we were having a really hard time honing down which cell types are expressing this enzyme, and traditional immunofluorescence imaging in aged muscle is kind of it's fraught with kind of technical issues such as autofluorescence and the accumulation of fibrosis and immunoglobulins in the tissue made it very difficult to be certain of what protein we were looking at and where it was localized. So we employed CODEX and CRISP to visualize this protein, and lo and behold, we were able to actually identify a very striking distribution of 15-PGDH in the aged muscle in multiple compartments. We were able to identify 15-PGDH positive macrophages that are not present in the young muscle, and we were able to find that a subset of aged muscle fibers actually upregulate 15-PGDH, and 15-PGDH is actually localized in a very interesting subcellular compartment in these aged muscle fibers. And both of these findings in macrophages and myofibers were validated by qPCR, and we're further exploring these in terms of mechanism of what is sequestering 15-PGDH, what is this subcellular compartment, and how does this relate to human disease. So this was a really great example of looking at molecular changes with CODEX and with multiplex imaging. Of course, we want to study with spatial data and multiplex imaging data, we want to study spatial relationships between cell types. And of course, for that, we must convert the images to single cell data by segmentation, quantification, and clustering. And by having better images that were processed by CRISP is really helping with both the quantification and clustering and segmentation. However, we're also making strides in improving this process. A common question is, how do you know you're clustering the right cell types and you're annotating them to the right extent? And you've seen from previous data presented by Kai that, with CODEX data, you might not necessarily get the kind of resolution in terms of clustering as single cell CITE-seq data or RNA data, which has more dimensionality. So we've tackled this from a couple of different approaches. First, we improved the segmentation accuracy with the help of Gary Nolan's lab. They've developed an algorithm to detect nuclei using neural networks instead of pure thresholding or watershed approaches. Second of all, we started quantifying protein expression in the nucleus versus cytosolic and membrane compartments. This is just simply using masks and expanding the mask of the nuclei and quantifying signal around the nucleus versus inside the nucleus. And that's actually able to overcome some antibody non-specificity issues and get us better resolution for nuclear versus cytosolic localization of proteins as well. Beyond that, we're now able to include pre-processing of CODEX data from across multiple experiments and automate this, the removal of autofluorescent cells and kind of debris that were left over from the neural network or segmentation of nuclei. And then, of course, improving the clustering of these user-selected markers. If we think about CODEX and CITE-seq, where we've chosen the antibodies that are included in the antibody panels, it makes it more robust for clustering. However, the clustering algorithms, even though we have clear kind of populations that should pop out, don't necessarily pick up on very rare cell types or resolve biologically relevant cell types. And so we've taken two approaches to a two-step approach to actually identify very cells with very high confidence stainings. We cluster off of those cells, and then we use a machine learning algorithm of label spreading or label propagation to then label cells with less confidence. And that's actually given us a very a large improvement in identifying cell types that were that the panels were designed to resolve. And so you can see that we can in a tissue like skeletal muscle regeneration, we've got roughly 30 to 40 tissues that we've imaged across multiple runs. And across these runs, we're able to identify about 25 to 30 functionally distinct cellular subsets. And you can see that with marker expression, each of these markers are able to identify the population of cells that they're looking for. And beyond that, by having spatial imaging data, we don't just have the protein expression data of each cell. We also have the morphological shapes and the spatial organization of the tissue. So what we've been doing is training neural networks to recognize features of the tissue, such as where the muscle is, where the motor neuron is, where the stroma is, and identify different types of muscle fibers, where the background is, and then basically feed this new information into the clustering algorithm as additional parameters that can be clustered on. And from this, we can generate really robust single cell annotations that can be used for downstream analysis. And this, I cannot stress enough how important cell annotations are, and comparing your annotations to what is actually in the image and what pathologists or human experts would actually annotate these cells. Because it's used for downstream analysis for spatial kind of relationships, it's really important to make sure that this step is as accurate as possible. So from our cell type annotation data, we've been able to generate a spatial cellular atlas of skeletal muscle regeneration. And also we've been able to compare what's happening with young and aged muscles. And across multiple experimental runs and multiple tissues, we get this very nice consistency in the changes in cell types that occur with injury and aging. Here you can see that M1 macrophages, which come into the tissue, infiltrate the tissue in order to clear dead muscle fibers. You can see that consistently. It increases at day 3 and then resolves by the later time points of regeneration, versus CD8 T cells, which occurs and comes into the tissue much later on. And even by kind of dimensional reduction and looking at projecting the abundance of cell types of each tissue into UMAP space, you can see that young uninjured muscles are clustered together, and all these different tissues are clustered together by regeneration time points. We even get almost a pseudo time kind of trajectory of very injured, resolving, regenerating, and almost recovered kind of time course here. And we can see that actually aged muscle is actually distinct in a distinct state, even just at the cellular composition. And we can see that aged muscles have more M1 macrophages and also even CD8 T cells kind of at the same time compared to young muscles. So how does these kind of very subtle changes in cell type abundance translate to the tissue architecture and how the tissue is organized? Well, looking at these cell type composition maps here, you might appreciate that young and uninjured and aged uninjured are not very different at the single cell level. However, by using neighborhood analysis of young and aged tissues, actually by looking at the compositions of cells and their surrounding neighbors, you can see that actually the difference between young and age is actually amplified. You see specific neighborhoods that appear with aged muscle that actually are reduced in young muscle. So this yellow here indicates this healthy muscle compartment that we've identified. And basically this is very abundant in young uninjured muscle, and then with injury it disappears and then slowly comes back with regeneration. However, in aged muscle, this compartment of interactions between healthy muscle fibers and M2 macrophages actually disappears. And this is possibly due to a M2 to M1 kind of polarization of the macrophage population in the muscle as well. We see more of the adaptive immune response neighborhood, which normally only comes up very late in regeneration, and aged muscles have a significantly more abundance of this neighborhood. And then we also see age-specific neighborhoods that are appearing where it's not really found in either normal young uninjured muscles or even recovering muscles. So this is suggesting that, you know, changes at the single cell level and the cellular composition level of the tissue is amplified by, you know, possibly signaling across cell types in the organization of the tissue. And so to try and get a sense of what could be occurring between these cell types and signaling, we wanted to kind of enrich this data set with transcriptome data. And so how do these cells change with age, and how do these how do changes in the transcriptome and cell-cell signaling, can it be actually translated to and used to identify pathways that go awry with aging. And so with the help of Akoya and actually BioLegend in providing some of the materials required for CODEX and CITE-seq, what we're able to do is perform CITE-seq experiments with this a largely overlapping antibody panel that we had used for CODEX. So just to go over kind of how these technologies work, it's actually very similar, but a kind of approach between CODEX and CITE-seq, where antibodies, the same clones of antibodies, are labeled with DNA barcodes, where in CODEX there's a short barcode, which during the imaging is detected by a complementary fluorescent DNA probe. Whereas once, you know, CITE-seq is performed on single cells, suspensions, and then has this longer DNA oligo conjugate, which has a poly-A tail that is captured during droplet-based single cell sequencing. And we opted for the Total Seq A panel because of its larger kind of panel constituency, and made it easy for the downstream process using the 10x platform. In terms of antibody panel design, we had about 36 CODEX markers and about 34 Total Seq A markers. With those 34 Total Seq, so CITE-seq antibodies, only about 30 were actually detected in our experiment. And there were, of those 30, there was about 20 overlap with the CODEX markers. Of course, there were some CODEX markers which were either intracellular, extracellular matrix, or not in the Total Seq A catalog. And for those, we were able to actually create a mRNA expression. We were able to use the mRNA expression detected by CITE-seq as a proxy for these molecules. One of the important kind of quality control that you can perform at this stage once you have your CITE-seq data and your cell type annotation of CITE-seq data here is that if you look at your embedding and clustering in protein space, you can see that you very nicely resolve all your different cell types. And, this is using a very high dimensionality with thousands and thousands of genes and the Total Seq A kind of protein panel, which will be able to distinguish all these different cell types. If you remove the RNA, what we're able to see is that, the panel itself was able to resolve the majority of these clusters on its own. And that's a really important metric to analyze, because, if your panel is insufficient in resolving all the different cell types here, it's going to lead to discrepancies in the data alignment and resolution of your data afterwards. We can see that the antibodies have a large congruence with the mRNA transcripts that were detected. So CD11B detected in the myeloid population was, in terms of mRNA, was also detected by antibody, CD31 mRNA and CD31 antibody, and then CD3 mRNA versus CD3 antibody. And so that tells you that the antibody panels that's being used is actually very consistent and able to detect cell types that express those genes. There were some subtle differences between mRNA antibody relationships. Here, if we look at Ly6G mRNA, which is really expressed only in neutrophils, compared to Ly6G antibody expression, you can see that the antibody actually labels all granulocytes versus just neutrophils for the mRNA. And so you will have slight discrepancies if you're trying to align mRNA data to antibody. On top of that, if we just look at, F480 gene expression versus F480 antibody, you can see that even though there is a large congruence between or a linear relationship between the gene expression and the antibody, there are some cells that, even though they don't express or didn't have mRNA detected, they are still positive for the antibody. And so this is important to remember if you're trying to just correlate antibody to mRNA directly. Already in the CITE-seq data, we can see changes in between the young and the age samples that were analyzed, mostly in that with the age samples, we observed reduced numbers of cycling, myogenic, and macrophages in the tissue. And then also we were able to see that there's an increased number of T lymphocytes and B lymphocytes, consistent with this bias of more adaptive immune response, maybe an autoimmune kind of response, which would be interesting for us to follow up. In aligning the data, you've seen algorithms such as Gluer and Stevia in this whole data alignment step. Here we're using a SCAMPy module called ingest, which takes the CODEX data with shared kind of features, shared feature space, such as the antibodies that are overlapping with CITE-seq data, and in addition to mRNAs that would correlate to CODEX proteins, and project the CODEX data into the CITE-seq latent space. So you're taking a lower dimensional data set and projecting it into a higher dimensional data set. And what we've been focusing on is taking this kind of projected spatial transcriptome to then model the spatial accumulation of growth factors, ligands, and then also predict juxtacrine signaling between cell-cell neighbors, focusing on kind of signals that require physical cell-cell contact. This is just to show you guys the metric of and quality of the data alignment. So by taking the CODEX cells and looking at their protein space and projecting it into the CITE-seq data, basically each CODEX cell is assigned a new cluster label. And when we do a confusion matrix between the CODEX labels that were manually assessed and validated versus the CITE-seq integrated labels, you can see that there's a good congruence. So B cells in the bottom left of this heat map correspond to B cells also identified by CITE-seq in the top of the y-axis on this heat map, and cycling cells correspond to cycling cells. Endothelial cells correspond to endothelial cells. However, we did have some mismatches that were identified. Interestingly, B cells express IgM1, IgMs, but in spatial data, we found that necrotic muscle fibers also accumulate IgM. And so you can see here that necrotic muscle fibers that were identified in CODEX also ended up being integrated as B cells here. So there are some kind of spatial and nuances and antibody kind of distribution nuances that with this data alignment. So for each CODEX cell, we then calculate a transcriptome using just k-nearest neighbors in this shared latent space that's in CITE-seq space. And basically, we use a distance-weighted, inverse distance-weighted mean for every gene that's expressed by the nearest neighbors. And we were actually able to get a decent projection or estimate of spatial transcriptomes. You can see here for integrin alpha-7 mRNA and CD11b mRNA versus the CODEX kind of protein data that we detected. So in thinking about cell-cell connectomics, traditionally people just take single-cell RNA-seq data or bulk RNA-seq data. They look at the expression of ligands and receptors in each cell type and then connect them together. You can do this with either looking at a single interaction or a single cell type in which ligands and receptors are connecting these two cell types. Versus, if you look at a single niche, so which other cell types are signaling to a single cell type, such as activated satellite cells highlighted here. This approach doesn't really consider the fact that cell types might never see each other in the tissue. And it doesn't really consider the fact that there are spatial kind of constraints to how signals kind of occur. If delta molecules are, and specifically notch signaling is a juxtamer molecule, if delta is expressed by cells that are never in proximity to notch expressing cells, then those signals cannot really occur. And so with space, we've really kind of focused on two possible ideas here, is that the spatial diffusion of paracrine signals, so growth factors are secreted by cell types, can travel a certain distance. And that distance, if you have accumulated kind of signals of lots of secretors within a recipient cell, that signal obviously will be higher. And so that's spatially determined. And because we have spatial information at the single cell resolution, we can kind of predict or impute where ligands may be and then calculate what that effect is on any given cell. And then we've also been able to use kind of very simple neighborhood analysis or adjacency analysis for each cell to identify possible juxtacrine signaling. And so combining what's already been done with databases and algorithms such as cell phone DB and ligand receptor interactomes, what we've actually been working on is kind of this method of estimating ligand diffusion. This required a customization of a novel database of ligands that basically, we can call upon to estimate diffusion constants. And then also, whether molecules signal specifically through juxtacron or paracrine means. And so at the end of all of this, you take your spatial data, you input it into the space pipeline, it identifies cell-cell interactions, it estimates the diffusion of the ligands, and then it filters out interactions based on receptor expression. And at the end, you get this sparse matrix of source target cell signaling. And then also you get a matrix of cumulative signals received by each cell. That's regardless of which cell type it's coming from. And so what you can do with this is, you can look at your CODEX image here. You can see that we have this injured skeletal muscle here with a large portion of dendritic CD11C positive dendritic cells here, surrounded by macrophages, and you can see kind of relationships with macrophages and cycling cells here with Ki67. And if we look at the space projection of where IL-1B might be in this tissue, you can see that it's actually accumulating around the dendritic cell and the injury site. And you can see that the receptor expression is also kind of correlated. So IL-1 receptor 2, which is the main receptor for IL-1B, is actually within the radius of this IL-1B kind of accumulation, whereas there are a lot of IL-1 receptor positive cells that are actually very distal to the source of the signal. So from this, we can obviously build kind of networks and graphs of how these cells signal to each other. And we can identify signals that are spatially restricted, such as IL-1B and IL-1 receptor 2, that are localized within the injury site versus actually peripheral signaling that we can see that occurs along the outside of the muscle. This is BMP2 signaling, which is quite interesting. And so, we now have a kind of database and unbiased kind of signals of roughly 2,000 receptor ligand interactions. And so you can also look at, which signaling interactions affect which cell type, and we can find modules of signals that are exclusive to any given cell types that could be targeted by antibodies or ligands or receptor blockade. And then also, you can see that using this method, we can identify stem cell regulators, myogenic stem cell regulators that have been previously identified in literature, such as EGF and OSM signaling in muscle stem cells. We can see sonic hedgehog and HGF signaling that affect also myoblasts, and then IL-6 and ampiregulin as regulators of differentiation. And of course, this is still kind of an ongoing iterative optimization of this space pipeline. But what I've shown you today is that a skeletal muscle is a manifestation of multi-scale and multi-omic process, including metabolic changes in prostaglandins, cell type composition changes, and tissue organizational changes. We can see that I've introduced you to a couple of technologies of how to study high-dimensional multiplex single-cell assays to understand these tissue-level changes, and then also integrating of CITE-seq data to enrich our understanding of how cell-cell interactions could be studied with spatial context. And so looking at space and using that as a platform to identify cell-cell signaling. So, and there, this has been a great kind of a large effort with many members of the lab. Adelaida, who was a previous postdoc in the lab working on prostaglandin signaling, James, Fiona, and Foster helped out with both programming and analysis of the tissue, and Jasmine, who is a MD student that visited the lab from Denmark, also helped with a lot of the tissue preparation, staining, and analysis. So with that, I'll take some questions.
Thank you, Will. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And please remember to take a moment to fill out our survey questions by clicking on the green survey icon at the bottom of your screen. We will now start the Q&A portion of the webinar. The first question is, are the neural networks used for identifying tissue features trained on RGB 3-channel images, or are they trained from scratch using more than 3 channels as input?
Yeah, so that's a great question. The neural networks are trained on the high dimensional data with individual antibodies as
Sorry, was there something else?
Can you hear me, Andrew?
I can hear you.
Yeah. No, I think that's it. Yeah, so using the neural networks, we're not aiming to detect every single cell type because that, you would have to train it on every single cell type. Here, we're identifying tissue features, which would then help the unbiased clustering, which is, unsupervised. And that way you can actually get cell types that you were not anticipating in your neural network training.
How are you doing cell segmentation?
So cell segmentation is being done on actually 2D slices, which, when we have the perfectly in focus tissue slice, we identify the center slice, and we do 2D segmentation of nuclei using a neural network, basically a ResNet trained, that's a UNet trained segmentation algorithm for nuclei, and that was developed by the NOLA lab.
Is data from the CRISP method publicly available?
We are going to make it publicly available when we submit this publication. For now, if you're interested in CRISP, we can send you the algorithm to try out, and we've seen very good results with multiple collaborators, and I think it's a really powerful tool. And it doesn't just work on multiplex imaging data; it works on bright field, it works on for stitching, it works on time-lapse data, and it works on traditional microscopy. It's really a powerful algorithm that can be applied to microscopy overall.
We have another question about CRISP. Could you describe a little bit more how tiling, deconvolution, and slide tilt drive tissue reconstruction in CRISP?
Yeah, that might take a little bit. The real power of CRISP is actually some of the math that's actually being done behind the algorithm. And basically, we've iteratively kind of optimized the methods for tissue tilt correction and assessment of which slice is most in focus. But really, the power is registering the images at subpixel resolution, and then stitching them and translating them at subpixel resolution. And what this means is, because the biggest kind of issues that we've seen with CODEX data and other 3D imaging data sets is that your Z resolution is usually lower than your XY resolution. And when you look for your best focus tile and you're trying to restitch your images, your Z plane will shift ideally not at a full full pixel step. And so by allowing subpixel resolution shifts, which is done all in Fourier transform frequency domain, what this allows us to do is estimate, subpixel shifts without losing that much quality, and basically you regenerate a new image that is now going to align with the tile that's next to it better. And beyond that, why chromatic aberration correction is important for this, it's not that huge of an issue with 20x imaging. However, you still do get, roughly maybe half a pixel difference between different channels. And when you're trying to stitch different images that have different kind of alignments, you're going to get a slight doubling, or basically your signal is going to get broader at 20x, but at 63x it becomes it makes the calculations not as good, because the centers of the images are the channels are perfectly aligned, but when you get to the edges of the image, there are subtle differences between how much you should shift your red channel and how much you should shift your green channel, and how much you should shift your blue channel and your far right channel. And so that kind of discrepancy makes the stitching and registration not as accurate. And so that's where CRISP really overcomes some of these limitations.
All right. Is there a minimum number of CODEX antibodies that should be used for correlation to work with the CITE-seq data?
Yeah, that's a great question. We've not really explored how few antibodies you can go, but this is dependent on your biological question. If you're only interested in a couple of cell types, you might be able to do that with fewer antibodies. However, if you really want to get the true power of single cell kind of data here with both CODEX and CITE-seq, you want to be able to capture every single cell type and really the biological heterogeneity behind that. And so that's why when I showed the quality control for CITE-seq, you really want to be able to see that your antibody panel is able to delineate the heterogeneity that is observed with RNA. That's a much higher dimensional data set, and that's got way more cellular heterogeneity. And so you should be able to use that as a metric to determine if your antibody panel is sufficient to delineate all your different cell types.
All right. And if people are interested in learning more about SPACE or perhaps using it, is it publicly available?
So SPACE is still in development, and if you're interested in collaborating with us, please reach out to me, and we can work on this, and we can try to analyze your data or put it through the algorithm. But it should be we're in the processes of optimizing both the database and kind of some of the math behind the algorithm still. But I'm happy to collaborate with people who are interested in getting a spatial understanding of cell-cell interactions and signaling.
All right, well, that's all the time we have for today. We'd like to thank Will Wang and our sponsor, Akoya Biosciences.